And I keep it really close to my footage, but
I'm happy to welcome you to our second session of Professor Josh Sandquist's quiz. Yeah, it is, right, hard to say. All about drugs. But before we get started, several announcements and reminders. Please silence your cell phones. If T-coil applies to you, please turn that on. After the class is over, the planning committee would really appreciate some able-bodied people to help put away chairs. Two of our strong guys are not here today, and so the two who are would really appreciate some help. Do that. There has been some question about Josh's class being, you can't find it on YouTube. That's because it's going to be put on sometime this week. We're going to do 2 classes at once, I think. So be patient. It will appear. If you had to miss last week, you will be able to find it. So with no further ado, I turn it over to Josh. Let's give him a welcome.
Okay, thank you. Let's check the mic again. Is it good? Okay, great. So thank you for having me back. Not running me out. Means I guess I did an okay job last time. We'll see how it goes this time. Have sort of a different angle we'll be looking at things today, but hopefully you find it interesting and informative. So this is week 2 of our bucket course talking about you and your drugs. Just a quick outline for today. I'm going to start with a really quick review of lecture 1. So I think it sets up what we're going to talk about today, but also there may be a few people who weren't here last week, so kind of get them caught up to speed. One of the things we talked about last week is that drugs work by physically binding and interacting with something in your body. So that was a general concept. We're going to develop that a little bit more today. And that's point 2 here, these cellular targets of drugs. We'll look a little bit how we can understand those. And that will allow us to then understand some features of how drugs work a little bit better. After that, because the natural extension of that idea that drugs only work by binding with something in your body means they have to get to that thing, right? So they have to get into the body and find that target. And so that's largely what we're going to spend our time talking about today. There's this concept, this acronym, ADME, absorption, distribution, metabolism, and excretion. So drugs have to be absorbed into the body, they have to distribute around the body. These are all things in order to get to the target that they have to bind to. And then we don't want them in our body forever, so eventually they get broken down through metabolism and removed by this process called excretion. So we'll think a little bit about that. This is something that's general to any drug you take, so that's what we're going to talk about. It's just general features of how drugs work. And finally, we'll have a little bit of broader discussion about some interesting things about metabolism. So that's where we're going today. So the review from lecture 1 that I promised. Last week we talked about the discipline of pharmacology broadly. We talked about it as the study of the relationship between drugs and your body. So we mentioned that drugs do things to your body also does things to drugs. And there's many different sub-disciplines, ways that people study drugs. So we talked about there's medical chemists who change the chemistry of a drug to make it work better. There's people who study the toxicology of the drugs. So there's all kinds of people who specialize in all kinds of ways of thinking about drugs to study how the drug works in your body. We also thought about exactly what a drug is a little. This term drug is a little loaded in our society, what people think it means. And so we thought a little bit about drugs versus vitamins. Or could we think of vitamins as drugs? And I think the short answer is yes. Most things we take that we eat or ingest, they're all, at some level, they're chemicals. And the chemicals work very similar ways inside your body. So artificially calling something a drug versus a vitamin, there's a long history to that, and the implications of that are some things are regulated and studied more differently than others. So we talked about that concept, what a drug is. But again, we're thinking mostly for this class of drugs as small molecules that we take in a purified form because we can better understand how they work if they're purified. We're going to get away from that a little bit in lecture 4 when we talk about other medical systems. And we went through a history of pharmacology, so all the way back to before research thought or any sort of organized research method, people recognize that when we eat stuff, it changes how our body functions, right? And then over time, we had an increased understanding of how our body works. So this is following on the heels of the physical scientists, chemists, people like, or physicists, like even back to Galileo. They started to put together these ways of investigating physical properties of the universe. And we thought, well, maybe we can understand the body in the same way. So people started to understand the body, how the body works. Improved chemistry techniques allowed us to purify chemicals. Improved other techniques allowed us to purify body components, and we started to understand how the chemicals in the body work together. And there was this concept of the drug receptor, which we're going to look at a little bit more today. And finally, we got all the way up to today. We have these clinical trials which have been designed to enable us to try to prove that a drug is going to both work and not be toxic. So that's a quick history of how we got to where we are today. But we're going to build off this idea today of the concept of drug receptors. And that goes back to what I started with. Drugs only work in your body by binding, physically interacting with something in the body. So one way to define a drug, if we ask the question, what are drugs, how do they work, is a drug is a chemical that modifies a life process. And I chose that word modifies specifically because drugs don't work by adding any new function to your body. Drugs can only work by modifying something that your body already does. It can increase a function, it can decrease a function to produce the result we want, but it can't produce a new function. It can't make something new happen in your body. So they bind things and either make them more active or less active or something. That's how they work. So as I said, most drugs work by binding some sort of receptor, some sort of cellular target. So I'm going to go through a little history again here, to just I think add some color to our discussion about how people began to understand these cellular targets. So first, I want you to think about and consider yourself as a multicellular organism. Your body is made up of all kinds of different cells. And if you think about you as your whole organism wanting to go through the world and act in a productive way, all the different cells and different things in your body have to work together, have to communicate well. So in a multicellular organism, all the functions that go in your body have to be coordinated in space and time. You wouldn't want, your pancreas secretes insulin, and insulin pulls the glucose out of your blood into your cells so they have that energy. If it's been a long time since your last meal, and you don't have very much sugar in your blood, you don't want your pancreas secreting insulin and trying to take that sugar out because it's already low. So that would create a problem. So that needs to be regulated in time so these things work together. So that's one concept. Now building off of that, so how does this coordination work? There's all kinds of signaling pathways that control these cell functions. So cells can create signals that other cells can receive, and then that allows them to coordinate their function. And there's many different types of signals so this is what's called a signaling pathway. A certain signal is produced that another cell can sense and respond to. There's all kinds of these different signals that exist in your body. And usually you can think of disease is one of these signals, these are not working properly. Something's over-signaling, under-signaling, the signal's not being received. And that's what we think of disease. Now, your body's not functioning normally because this information's not getting where it needs to go in the right way. So as a result of that, the cellular molecules, the cellular proteins that control these signals are often the targets of drugs. We want to fix the signals that are going on in our body, basically. So let's think a little bit. So I'm going to try to use some examples. It's a little bit of an abstract idea, so we'll kind of develop that a little bit. I'll make it less abstract, a little bit more concrete for you, hopefully. And I'm going to tell you a little story about, I've only read this person's name in print, so Charles Edward Brown Sicard is how I would pronounce it. He was a really quite accomplished, famous scientist in the late 1800s, published hundreds of papers. He was a successor to Claude Bernard, we talked about last week, as the head of the College de France, contemporary and friend with Darwin and Thomas Huxley. So a very accomplished scientist. I'm going to read you a little excerpt from one of his papers that generated a little controversy in his day. So the title of the paper, The Effects Produced on Man by Subcutaneous Injections of a Liquid Obtained from the Testicles of Animals. So he was deriving this liquid from, I think it was young calf testicles, I think is what it was, and he was injecting this under his skin. So this is his report on this, a part of it. He says, I am 72 years old. My general strength, which has been considerable, has notably and gradually diminished during the last 10 or 12 years. Before May 15th last, I was so weak that I was always compelled to sit down after half an hour's work in the laboratory. Even when I remained seated all the time, I used to come out of it quite exhausted after 3 or 4 hours of experimental labor. He goes on to describe this a little bit more, and he sets up the idea how he was going to do this injection. So he said, the day after the first injection, and still more after the 2 succeeding ones, a radical change took place in me, and I had ample reason to say and to write that I had regained at least all the strength I possessed at a good many years ago. So he just goes on about that a little bit more, and then he wants to measure it. So he says, I have measured comparatively the effects before and after the first injection. I measured the jet of urine in similar circumstances, i.e., after a meal in which I had taken food and drink of the same kind in a similar quantity. The average length of the jet of urine during the 10 days preceding the first injection was inferior by at least 1/4 of what it came to be during the 20 days following. So we have to measure what we have available, right? So he wanted to find a way to accurately measure the effect on his body, and after measuring that he could pee further, he said, it's therefore quite evident that the power of the spinal cord over the bladder was considerably increased. So, he published this, created quite a stir. A lot of people, some scientists thought that this was very interesting. As I said, he's a very well-respected person. Some people objected to these things on moral grounds. It seemed weird to inject animal fluids into your body. How's it different than eating animals? I don't know, but some people found it different. You might imagine this became, over time, people sort of warped things as sort of like a youth elixir, and so then other people started trying to do the same thing, and it created some unsafe situations. And over time, Brown Sicard started to maybe wonder had he made a mistake in publishing this. Nevertheless, a lot of researchers saw the potential of what he discovered here. And so, while organotherapy was controversial, began to gain acceptance, people studied it more. By the way, Brown Sicard wasn't the first to do that. So if you go way back to the ancient Greeks, probably before, they were eating animal organs to produce different effects. Supposed to eat bull testicles for virility, or women trying to get pregnant were supposed to eat rabbit reproductive organs. So they thought that there was, how much of that is based in evidence or mysticism, we don't know, but this wasn't the first example of people using animal organs to produce an effect on our body, right? So people began to, even though it was controversial, people continued studying this, including Dr. George Oliver in England in the late 1800s. He discovered adrenaline. I'm going to read to you, there's a really interesting account of this discovery as well. So this is someone's account of the discovery. It says, Dr. George Oliver, a physician at Harrogate, employed his winter leisure in experiments on his family. Using an apparatus of his own devising clinical measurements. In one such experiment, he was applying an instrument for measuring the thickness of the radial artery. So he wanted to measure the arteries in his arms, measure how thick they were. And having given his son, who deserves a special memorial, an injection of an extract of the supraadrenal gland prepared from material supplied by the local butcher, Oliver thought that he detected a contraction, or, according to some who have transmitted the story, an expansion of the radial artery. Whichever it was, he went up to London to tell Professor Schaefer, a well-known researcher in this field, what he had thought that he had observed, and found Professor Schaefer engaged in an experiment in which the blood pressure of a dog was being recorded. He found Professor Schaefer was not unnaturally incredulous about Oliver's story, very impatient at the interruption. But Oliver was in no hurry, and he urged only that a dose of his supraadrenal extract should be injected into a vein of the dog when Schaefer's own experiment was finished. And so, just to convince Oliver that it was all nonsense, Schaefer did indeed gave the injection, and then stood amazed to see the mercury mounting in the arterial manometer till the recording float was lifted almost out of the distal limb. So it had this huge effect. Surprise. I gotta do more experiments on my children, I guess. The short takeaway of this, right? I don't think my wife's gonna go for that. Probably a smart idea to not do that. So, we discovered adrenaline, right? So these are experiments, and we're going to stick with adrenaline for a little bit, because I think it's something that's going to help us unpack some of these ideas. So adrenaline, some of you have probably heard of adrenaline as the mediator of the fight or flight response. It's released from the adrenal gland, which is just above your kidney. We'll look at that in a minute. Goes into the bloodstream. This happens in the anticipation of danger. So it's supposed to promote a whole body response that helps you fight or flight the danger. I think it's probably demonstrated better in this image. Now it turns out this is actually, evidently they're friends, and the guy just wrestling with his friend the tiger. But to me, in that situation, my adrenaline would probably be pumping. And what does adrenaline do? Adrenaline has all kinds of effects. Here, go back to this here. So this is your kidney. These are the kidneys, the urine collecting duct that goes down to the bladder. And sitting on top of the kidney are these adrenal glands. So this is where the adrenaline is produced. So you sense danger, adrenaline is released into the bloodstream here, and then it's quickly distributed all over the body. So adrenaline goes to different body parts, and it binds. So this is one of those signaling molecules we talked about. This is how the body coordinates functions inside the body. So adrenaline is a molecule released into the blood, floats around, and it has to bind to its target, which we call receptor. And it binds this receptor that lives on different tissues to produce different responses. So, one of the responses to adrenaline is the pupils dilate, to let more light in so you can see better the danger that you're trying to fight or fly away from. The bronchi, the airways, open up to let more air come in. The heart rate quickens, so blood moves around faster so you can move the oxygen around so you can power those muscles. Some blood vessels dilate or open, so the blood vessels in your arms and legs, they actually open up to let more blood in. But the blood vessels in your abdomen, you don't need to be digesting your meal at this moment, you need to be fighting or flighting, right? So there's less blood going to your internal organs and going out to your arms and legs. Energy stores are mobilized. So adrenaline, a single molecule, makes all these different things happen by binding to its receptor in different places in the body. So just another visual representation of how this works. Imagine this is a cardiac muscle cell, so muscle in your heart. You're going to want it beating faster so you can, your blood rate can go faster. So if this is the cell, the receptor that we're talking about, the target that adrenaline is going to bind to, it's called adrenergic, adrenaline from adrenaline. The adrenergic receptor is a protein that lives on the surface of the cell, and half outside the cell, and half of it is inside the cell. So it spans across that boundary. So if you have adrenaline floating around in the bloodstream, your adrenal gland released that adrenaline, now it's floating around, and it bumps into the heart, comes by the heart. The adrenaline molecule has a very complementary shape, and it will bind and interact with that receptor. Now what this does is it sort of changes that, and you can just think of this simple way, just think that it turns this receptor on. We're not going to go into all the details here, but that creates a signal that goes into the cell and tells the cell to contract and increase the heart rate. So this happens by this molecule floating, binding this receptor, has to physically bind. It changes, so it turns that on, like a switch. Has anyone heard of propranolol? Or any other drugs with all at the end? They're all a similar sort of category of drug. These are what are called beta blockers. So these adrenergic receptors we just talked about, there's different flavors. There's alpha version, a beta version. So propranolol, so it's called a beta blocker. Oops. Where did I go? It blocks the beta version of these receptors, so adrenaline can't bind. And this is used to affect things like hypertension, high blood pressure. So blood pressure, you can think of, if your heart is pumping blood, you have all your blood vessels. If you leave your blood vessels sort of the same diameter, but your heart pumps faster, pushing more blood in there, the pressure is going to increase. Or if your blood vessels contract down to a smaller area and the same amount of blood is going in, the blood pressure is going to increase. So we can change blood pressure by either slowing down the heart or causing those blood vessels to open up a little bit more. And all these things are controlled by the adrenaline that we just talked about. Not so we could develop drugs to fix your blood pressure. Remember, it was just part of this fight or flight response, but we're tapping into that to control what we want to control in terms of your blood pressure. So, to visualize this here, propranolol would be a molecule that part of it looks a lot like adrenaline, so it can still bind this receptor. So it'll go in and occupy that spot, but it doesn't do it for the receptor and doesn't turn it on. So it binds there. It won't tell the heart muscle to contract. But now, if adrenaline tries to bind, it finds that place occupied, so it can't bind. So this is why it's a blocker. It blocks those receptors so adrenaline can't bind. Yes? Oh, yeah. Go ahead. Yeah.
Going back to the kidneys with the adrenaline gland on top of the kidney. When a kidney is replaced, kidney transplant, what does that do to the adrenal gland on top? How would that affect a new kidney? Is the adrenal gland damaged? What happens there?
Yeah, so the question is, if we did a kidney transplant, how would that affect this adrenal gland? And the shorter answer is, I'm not 100% certain if the adrenal gland is brought along with the kidney transplant or not. We have two, but I think the body's able to adapt. And so if it had only one adrenal gland, systems would sort of reset in order, so that's still sufficient. I think people can get by with one kidney in that way. Maybe the adrenal gland is taken along for the ride. I'm not 100% certain. Yes?
Okay, if you take a medication and it says these complications can occur, would that possibly affect your adrenal gland, or what would it be?
Yes, so the question is, if you take a medication like propranolol and it says these side effects can occur, is that occurring because it's acting on the adrenal gland or something else? Well, the issue there is that, as I said, adrenaline binds receptors in your eye, in your airways, in your heart. So if you take propranolol, your target's really the heart because you want to slow down the heart rate. It might still bind to those receptors on the eyes or in other places. Sweat glands will also increase sweat, and so sweating can be a side effect, or lack of sweat can be a side effect. So this is a non-desirable effect because we don't really want to affect those things, but it's not what we would call an off-target effect because the drug is still binding things it's sort of designed to bind. It gets a little, we can try to make drugs that will only bind the ones on the heart and not other places, but we're limited in what we can do there. So that's the basis for some of these effects. We just know if you take propranolol, it'll probably cause some of these other things just because it's going to bind in those places too. Does that answer your question? Okay, yes.
What about cerebral hemorrhage when these are introduced in the kidney?
So the question was about cerebral hemorrhage. Could that be a possibility? I don't know. I mean, if you know, is that something you've heard as a possible side effect of these, or you're?
In our family.
Okay. So yeah, I'm not sure if there's. I don't know a lot about all the specific effects of different drugs. Many things you wouldn't expect to affect the brain as much because it's something we're going to talk about in a little bit. There's an extra barrier in the brain that makes it harder for chemicals to get in there. But it sounds like it's something that's really happening, and I can't give you a reason why right now off the top of my head. Yes?
This is a strange question, but I was wondering, if you're having a bad dream, does the adrenaline gland ever give off adrenaline? And
Yeah.
cause high blood pressure in that dream after you have the dream?
Yes, so the question was if you have a bad dream, is adrenaline released? The answer is yes, because you can see if you've ever woken up from a bad dream, you have a lot of the symptoms, right? Your heart's pounding fast, a lot of times you're sweating. And it's not necessarily because you have too many covers on, it's because that's the response to adrenaline. So your brain has triggered that adrenal cortex to release the adrenaline because it was scared. Even though it wasn't a real threat, it still receives the fear in the same way. Oh, these are all great questions. Thank you all for all these good questions. So adrenaline is a target, or this pathway, this signaling pathway we talked about, target a lot of different drugs for a lot of different purposes. Eye drops that you take to the doctor can work on the same pathway because it dilates the eye, so there's all kinds of different things. So I hope this illustrates what I mean by a drug having to bind a specific target in order to work. And it doesn't create a new function, it either increases or decreases a function that already exists in the body. Okay. The other thing, this isn't the only receptor that lives on this cell. There are other receptors, but they look different. So propranolol is designed such that it'll only bind this one and not bind this one. If it bound all these receptors, it would block all these different signals, and then you'd have a whole array of problems, right? So that's what the chemists do. They try to design these drugs so they bind only the specific thing you really want it to bind to. Here's another example of that's less cartoony. So BCR-ABL is a mutated form of a normal enzyme that lives in your body. And what this enzyme does, the normal version, tells your cells to grow and divide. We're all adults in here. So as an adult, most of the cells in your body are not growing and dividing. Kids, they have a lot of growth, a lot of vision. But as an adult, you sort of reached your final size. So not very many of the cells in your body are actually growing and dividing, only to replace damage, mostly. There's a few exceptions, places like hair cells and the cells that line your intestines. Those are growing and dividing pretty regularly. So this thing normally tells the cells to grow when needed, but most of the time it's turned off because we don't need a lot of growth. Now, there are cases where this is mutated and it's turned on all the time, and that leads to cancer. Cancer is uncontrolled cell growth. You have cells growing and dividing and multiplying when they shouldn't, and that's where you get a tumor. It's a large mass of cells that shouldn't be there. So this particular mutation is found in leukemias, specifically CML. So every single patient who has CML has this mutation. Other leukemias like AML, almost no one has this mutation. So it's a very specific mutation to that type of a cancer. Now as you can see, this drawing here is a representation of the shape of the enzyme. There's something called X-ray crystallography where they're able to use these powerful X-rays to determine the shape of proteins. And so this is a drawing representative on what the protein actually really looks like. And you can see there's this, cleft or this cavity kind of right here in this spot. Well, there's a drug called imatinib, or maybe people have heard of Gleevec. It's a very famous drug because it was the first example of rational drug design that was successful. Most drugs, we sort of just find them by, we have a whole library of thousands of different drugs, and we say we want one that binds this protein. You just kind of run through a screen. Does this one bind? No. Does this one bind? No. And you kind of search for one that binds and does something. Well, Gleevec was one that we sort of designed rationally to fit this protein because we knew this protein was the causative agent of the cancer. So it fits right in that little pocket there. And if we look at it differently, the purple area here represents where the drug, imatinib or Gleevec, is. And all these structures here are pieces of the protein that live around it. So you can see this thing fits just perfectly in that void there. And more than that, there's certain atoms that are part of this chemistry are in just the perfect place so they can form a bond with different things that exist in the protein. This is what the chemists do to try to make sure that imatinib will only bind this particular protein. We don't want it binding the normal version of this because then it would turn off those signals in your normal cells. In this way, it binds only this mutated version, which only lives in your cancer cells, if you have that particular form of cancer. So again, this is just an example, another example of how drugs are designed specifically bind something in the body, and they work by either increasing or decreasing the function of that thing that they bind to. Most drug targets are proteins, but just to sort of give a counterexample, they're not all proteins. So I was trying to think of one. Cholestid is an example of a drug that's a lipid-reducing, a cholesterol-reducing agent. And so your gallbladder takes cholesterol from your body, turns it into these things called bile acids, and then secretes those out of the body into the intestine to help digest foods. So that sort of pulls cholesterol out of your body. What cholestid does, it just binds those things in the intestine, prevents them from going back into the body, and they just come out the rear end. And so it draws the cholesterol out of your body this way. So it doesn't bind to a protein, doesn't even have to really go into your body, so to speak, because it just stays in the intestine. This, by the way, is essentially how oatmeal works. That's how you take oatmeal to reduce your cholesterol. Same idea, it binds those things and keeps it in the intestine rather than being absorbed. So it reduces your cholesterol that way. So we have these drugs, and the idea is, if we're going to imagine understanding what we do now about how drugs work, an ideal drug would be one that produces the action we want, to produce the effect. It would have no side effects. Like the ideal work, how many times does the ideal actually exist? We would hope that it reaches its target, wherever that is, whatever that is, at the right time, at the right concentration. You wouldn't want to take an aspirin and have it take 6 hours to get there, because your headache would probably naturally abate before then, right? It would remain there at that site, doing its interacting for a sufficient amount of time to produce the effect. Then it would rapidly and completely be removed from the body, so it's no longer around. That would be an ideal drug. So these features here, these last three, gets to the target at the right time, right concentration is removed. These are all under the domain of that ADME that we talked about. So different drugs have different mechanisms of binding their target and producing effect, but all drugs are subject to this getting into the body, distributing around the body. So this is where we're going to unpack these concepts a little bit here. So we usually take a break about 9:40, is that right, 10:40? Okay, a couple more minutes here. So, A is absorption, how drugs get into the body, absorbed into the body. D, distribution, how they get around the body, where they need to go. Metabolism, the body breaks down, chemically converts these drugs for different reasons. We'll get into that a little bit. And then excretion is removed from the body, getting the drug out of the body. Start with absorption. Now, how a drug gets into the body varies a lot depending on how you take the drug. There's all kinds of ways. Oral is the most common, you swallow a pill. But drugs can be taken by injection. You can either inject into the vein, into the muscle, like anyone got a flu shot, yeah. Subcutaneous is under the skin. Not very many of these, but there are some that are injected into the artery rather than the vein. Some drugs are taken rectally, some drugs are taken under the tongue, transdermal patches, nasal sprays. Does anyone have asthma? They've taken it through the lungs, like inhaled something in the lungs. That's pulmonary. I always never say this word right. Ophthalmic. Eye. So there's other ones I haven't talked about, like an epidural into the spine. So there's all kinds of different ways. But by far, in a way, oral is the most common. It's the safest, most convenient, most economical. The advantages are that it's easy to take a pill, not invasive. You don't have to stick any needles through the skin, which could cause an infection or something. You don't have to go to the hospital to have it done. You can just take it at home and take it. Some of the disadvantages are absorbing drugs that are taken that way orally can be a large barrier for a lot of drugs. So a lot of drugs have limited absorption. But it's something we try to deal with because it's so easy and convenient to take pills. Some drugs can be broken down in the stomach. It also requires patient cooperation. If a patient forgets to take their pill, or takes too many, or takes it at the wrong time, or eats when they're not supposed to, it can all limit the effects of drugs. So these are all trade-offs that we make. But again, oral is the most common way. So that's the way we're gonna think about absorption. So for orally administered drugs that you take via mouth, this is a pill, but it could also be anything like there's a liquid you drink or something, anything that goes into the stomach. Most absorption actually occurs in the intestines, not in the stomach. So the stomach's job is to break things down. When you eat, the job of the stomach is to physically grind it, and it uses acids to break all this stuff down. And there's a little valve here that lets the things out of the stomach, and it goes into the small intestine, and this is where the vast majority of stuff is actually taken into your body. We'll explain that in a second. It'll go through the small intestine and then go through the colon, otherwise known as the large intestine, and out the rear end. So this small intestine is a tube, it's a really fancy tube. If you think of yourselves this way, we are all just really fancy tubes. Because we have a tube on one end, an opening, the tube on the other end, and then just stuff hanging off the tube, right? So we put stuff in our body that's connected to the outside and has to be absorbed through the tube to get into our body. So the inside open area of this tube, of the intestine, is called the lumen. I'm going to refer to that again. So that's just inside the tube. And you can see here the small intestine is connected to the bloodstream. If you open this up and look at it, you'll see that the inside of this tube is not smooth. There's all these folds. And then on the folds, if you zoom in a little bit more, on the folds there's even more little folds. And each of these folds, called villi, have an artery and a vein inside them. And there's only one cell layer between the lumen on the outside and the blood layer, the blood on the inside. And if you zoom in at these individual cells, there's even more folds on top of that. So there's folds on top of folds on top of folds, increases the surface area here. So the surface area of your intestine is something like 200 square meters, which is about half of the end zone of a football field. So if you took your intestine and spread that out and made it flat, that's how much area it would cover. So it gives a lot of area for drugs, for things to be absorbed. This is one of the reasons it's so good at absorbing things. There's just so much area there. The other thing is the high blood flow. As soon as something gets absorbed in, it's captured by the blood and taken away. So it doesn't have a chance to accidentally go back out. So the intestine is designed through evolution to be an expert at absorbing things. So let's take our break there. We'll come back and think a little bit about what it means to be absorbed, why it's so challenging. And that's what we'll pick up when we come back.
So that Josh can get started again. But before he does that, this pair of gloves was found at the beginning of our time together on the floor near the coffee. Belong to anybody here? Yeah? Okay, all right. Well, thank you.
They're black and gray.
Are they? Thank you, Sue.
You don't want to leave today without your gloves, man.
Good.
All right, well, welcome back. We'll talk a little bit now about how the caffeine you just drank or the food you just ate gets into your body. It's a very practical concern for us right now. Luckily, you don't have to think about it, it just happens. So much of our body. It's magical that way. Okay, so we're going to talk about this idea of absorption. You take a drug, the pill goes into your stomach, gets broken down, moves into the small intestine. How does it get from the empty space inside that tube, the lumen, how does it get into the bloodstream and around to your body? Well, as I mentioned here a minute ago, if you remember, this little wrinkle here is surrounded by cells and it's only one layer thick. A lot of places in your body may have multiple layers of cells stacked up on top of each other, but this is only one cell layer thick, which is useful because that's relatively thin by certain standards, so it makes it a little bit easier for stuff to go through. But it still represents a significant hurdle for drugs. So if we, I blew this up and I drew this cartoon, so these are the cells you can imagine. This is called an epithelium, it's just a term for the type of cells that line the intestine or different organs. So I drew this, and these are all those little wrinkles. So the space above here, is the intestinal lumen. So you imagine that's the hollow space inside of your intestine. So the drug has to go through the cell layer to get on the inside where the capillary or that blood vessel will be, so it can get into your blood. So this blue thing here, this little blue, whatever that is, hexagon, is the drug. And one thing I'll mention, these blue lines here, these represent really tight connections between the cells. So these cells that line your intestine, they still have to form a barrier so that bacteria and things that are on the food that we eat, everything's going to have bacteria on it. They can't get into your body too. So even though these are individual cells next to each other, they're kind of almost sewn together. So things can't go around the side of them. So the drugs really have to go through those cells there. Now this is a trick for this drug, so I'm going to do something like that and float away in the bloodstream. So this is a trick for the drug because cells are basically just these things that are surrounded by what we call a membrane. So there's one membrane layer it has to go through on one end, then it gets into the cell, moves through the cell, then it has to go through the membrane again. So it has to pass this membrane 2 times to get into the body. This is the challenge for the drugs, getting through that membrane. So outside of the cell, in the lumen, is sort of like a watery environment, an environment that's composed largely of water. Same on the inside of the cell. Even though there's lots of proteins and other things in there, it's not exactly like a soup. It's more like a really thick gel or syrup, like the thickest molasses you might ever imagine. It's still water-based. It's really thick. It's got all these proteins and things in there, but it's still water-based. So you have these two water-like environments, but the membrane is a lipid-like environment. So this would be like the fat that forms that really thin layer on top if you let it cool after you've stewed a bone or something, and there's some, so the fat, the lipid, or fat, and the water don't mix, right? They separate from each other. And these have very different chemistries. So something that's happy inside water is generally not very happy inside of a lipid. Like oil and water don't mix. So this drug ultimately wants to get into a cell, which is a water-like environment. So it has to go from a water-like environment to a lipid environment back into a water-like environment. And this is the struggle. If it's happy in one, it's usually not happy in the other. So this is the fine balance. This is why it's hard for drugs to get in. So a drug must be able to be soluble or be able to dissolve or break down or live happily in a water environment, but it has to travel, so it can get around the blood. All the biological tissues are everything's water-based except these membranes. So it needs to be happy in that water environment, but also has to be happy enough that it can go through these lipid-based membranes. And so what we want to know when chemists are designing drugs, they want to know the ability of a drug to dissolve in a lipid when the water is also around. So how do we do this? Well, they set up a very simple experiment. They take like a beaker, put some water in there, and they put a chemical called octanol, which has a lot of characteristics like fat, so it's like a lipid thing, and they don't mix. So if you set them in there and just let them settle, they'll separate into these two layers, kind of like the oil floating on top of the water. And then you throw in some drug that you're testing and wait a while, and you measure how much of it is up here in the lipid layer and how much of it's down there in the water layer. And then you just do math and you do a ratio. You say the concentration in the octanol or the lipid divided by the concentration in the water. And we just know, over years of testing this, we want this value to be a certain number to where it'll be happy in both environments to a certain extent. If the number's too high, almost all of it's up here in the lipid and not in the water, it's not going to work because it can't get around the water tissues, or the water inside the tissues. If it lives all in water and not much up here, it's never going to get into that membrane and go through. So this is a number that chemists will make changes to a drug and they have to check this number, this partition coefficient. So the problem is a lot of drugs, maybe we'll make them, if we have to make a change to its chemistry to make it a good number here, then maybe it no longer works on the biological target. So, we can fix the absorption problem, and then it's not going to do what it needs to do. So this is the balance, and this is why not all drugs are capable to be taken by pills. You just can't make them soluble enough that they'll be absorbed and then still function. So these are things you would have to take by injection. So an example is, I don't know if anyone's heard of Taxol. It's a common chemotherapy for cancer. It's just this really large beast of a molecule. This can't be absorbed, so it has to be done by injection. Okay, so we got the drug absorbed into the body. So we designed it such that now it can go through the membranes, but it's still happy in water. It goes into, so it passes through this tissue layer, and then it'll get to the blood vessels here. Okay, that's the target of that. So now how is the drug going to move around the body? Most commonly the target's not just right around the intestine, it has to go somewhere else to work. This is a diagram of the blood vessels in your body. So we have the blue side, which are all the veins. These are things that are returning blood back to the heart. The red side are the arteries. These are the vessels that take blood away from the heart. So you have blood, let's say it's coming back to the heart. The heart pushes it up here to the lungs, where it collects a bunch of oxygen that you breathed in. And then now that blood with the oxygen in it leaves the lungs, goes back to the heart, and the heart pumps again and then pushes all that blood with the oxygen out to your liver or your stomach or all your different parts of your body. So these large, like the aorta, it's probably something you've heard of, a large artery. These large vessels here are called arteries. They're designed as sort of like the interstates that are designed for fast travel. Stuff's not really going in and out of them very much. It's mostly just to move it somewhere else. We have these smaller, we'll talk about these more in a second, these smaller vessels called capillaries. They're designed differently such that stuff can come in and out of the blood vessel more easily. So you have these large arteries which carry the blood, and then let's say here it gets to your liver, then these vessels fan out into the little capillaries. The blood then bathes the liver, and stuff's able to go in and out as needed, and then it goes into the veins, which again don't have a lot of exchange in and out, and they're designed again for rapid travel back to the heart. So these things here are called capillary beds. They surround the tissues. This is where the blood goes, it gets out, carries the oxygen to the muscle or wherever it's trying to go. So what I want you to note here, and we'll come back to this will be important in a second, is that most things, the blood comes from the artery, goes to the capillaries, right into a vein. The liver, artery, to the capillaries, to the vein. The stomach and the intestines are unique in that the blood comes from the artery, goes to through the stomach and the intestines, then rather than going right back over to the vein system, it goes up to the liver and then goes back. So there's an extra little jaunt that the blood takes after it leaves your intestines. It goes through the liver first. That'll be important in a minute. Here's just a diagram showing you the difference between these large vessels. So they have a lot of muscle around them. That's what I said, that's where the adrenaline acts. It'll cause that muscle to contract to make the blood vessel more narrow, or expand to make it wider. This is how one way your body controls blood pressure. So it's got all these layers of stuff, this membrane, this muscle, another outer coating. So it's hard for stuff to get in and out of these vessels. So again, these are designed to move blood long distances. These smaller vessels are the capillaries. They have just this minimal layer of cells that form the tube, and then one sort of membrane holding things together. And then if you look at them even a little bit more carefully, there's a couple different types of capillaries that are more leaky than others. Some are pretty tight, some have little holes in the cells, some have these humongous gaps. So something like the liver, there's a ton of exchange in and out. The liver's designed so things can go in and out of those capillaries very, very easily. So this is all, depends on the tissue, depends on the organ, you want more or less blood exchange coming in and out. So these are the types of capillaries that are in the tissues. One special place is the blood vessels that supply the brain, which aren't shown in this diagram. So here's the lungs, and you would have some sort of blood vessels going up to the brain. The brain has a very unique type of capillaries. Most chemicals are not able to get into the brain. This is something called the blood-brain barrier. So there's an exceptionally tight cell layer in those capillaries, and there's also enzymes in those cells that either pump stuff back out or break it down. So the brain has a really tight barrier to really tightly control what can go in and out. A lot of drugs, even if they can get through the intestine into the body, still can't get into the brain. They have to be even a little bit more lipid soluble to get into the brain. So that's an extra challenge for drug designers. So the brain is a very special case of these capillaries. So I had a question earlier in the audience, from someone in the audience during the break that asked, how does a drug know where it needs to go? And the answer is, it doesn't really. It doesn't go somewhere in particular. If you have a drug that's going to act in the heart, it's not like it only goes to the heart. When the drug gets absorbed and goes into these blood vessels, it's mixed in the blood and it's just kind of spread out all over the body. But maybe only the thing that we want it to bind to only lives in the heart. So even though the drug is down here in the kidneys or the lower body, it's there, but it's not doing anything because its target isn't there. So the distribution just spreads it everywhere, and then we hope it acts with its target. Now, sometimes there'll be other things that'll bind, or in the case of, like we talked about the adrenaline, there's kind of these targets all over the place. So that's where you start to see more side effects. It's binding stuff, it's binding on the heart where we want it, but also other places too. Okay. So, that's the drug getting in, the drug distributing. Let's move along here. Now, metabolism, the breakdown of drugs, and the elimination, I think is the most interesting. So in order for a drug to get out of the body, it can be excreted or metabolized, or this is the sum of those things. Excretion, by definition, is a drug that the kidney can remove the natural form of the drug. It doesn't have to be metabolized, doesn't have to be broken down. The kidney can just remove it. Some drugs, though, aren't very good at being removed by the kidney, so they have to be metabolized by the liver first. Oh yeah, yep.
What about drugs that are not taken orally in any form, but are rubbed onto the skin?
Yep.
Right at the spot of what is sore. How does that work?
Okay.
Does it eventually go through all the body as well?
Yeah, good question. So the question is, what about a drug that you have like in a patch or an ointment or something you're rubbing on the skin? Does that go everywhere? The answer is sort of yes, but the impact of that sort of depends. So let's say there's a little artery here that goes out, because all parts of your body at some level have a little blood vessel supplying the blood, right? So if you put something on your arm, some will probably be absorbed through and eventually get into the bloodstream. But typically, if you want it to act only there's such a small amount that it'll be a high enough concentration to be functional there, but when it gets into your blood and distributes out through all this liquid, now it's so diluted that the concentration is so small that it won't produce any effect. But some things we will take transdermally, like nicotine patches or other things. We put a high enough concentration in there that it'll be absorbed, and then it eventually makes its way into these blood vessels and spreads out. So you can use it as a way to treat the whole system as well, but a lot of times those things are meant to only act locally, just because you wouldn't get enough in. They don't even put enough there. Thank you. Good question. Okay, so most things are removed from the body via the kidney. There's other ways, but that's the vast majority. Some things the kidney can just remove them as they naturally come in the form that they're normally in. Some things have to be metabolized or modified by the liver before the kidney can remove them. So this is just a diagram showing that. This is a drug. The water-soluble ones, again, the ones that are more water-soluble typically don't have to be metabolized. The kidney can just remove those. The ones that are a little bit more lipid-soluble, often the liver has to metabolize them, change them to make them a metabolite, so that's a new product of that. That's a little bit more water-soluble, so the kidney can remove it. So metabolism relies on these enzymes, which can take an unexcretable drug, or in this case, the enzymes didn't evolve in our bodies to remove drugs, right? We didn't always take drugs in evolutionary times. But there's all kinds of foreign molecules that our body were exposed to. Biological stuff from what we ate, or just chemicals that existed in the environment, or in the water we drank or something. So our liver evolved to help get rid of those things. But they still act on the drugs. So the xenobiotic is just another term for foreign molecule, something that's foreign to the body, in which case the drug is a xenobiotic. And enzymes then convert these drugs into some metabolite that's usually better excreted by the kidney. Metabolites are sometimes inactive, so if the drug is metabolized by the liver, sometimes it's turned off, it can no longer function because it's been chemically modified. Sometimes that's not the case. Sometimes they're modified, but they're still active. Sometimes they're modified into metabolites that are now toxic. So the original version wasn't toxic, but the liver converted it into something that the kidney can remove, but that new thing is also toxic. So there's all kinds of potential outcomes here for what the liver can do. So I want to give a couple, I hope, interesting examples for how the liver works, using some things that we might be familiar with. So oxidation is the type of modification, doesn't really matter to you for us, for our purposes, what that means. That's the most common type of metabolism reaction that occurs. So alcohols, there's a whole category of alcohols. Ethanol is the most common one to people, so that's what's in beer and wine, things we drink. So this is the chemical formula for ethanol. Now, when ethanol is metabolized by your body, there's an enzyme called alcohol dehydrogenase. What it does is it removes those two hydrogen atoms there and puts an oxygen atom there, creating a new chemical called acetaldehyde, or acetaldehyde. Sorry. So it's no longer ethanol, doesn't produce the things that ethanol does. But that's not the end of the road. There's a second enzyme called aldehyde dehydrogenase, which then takes off this hydrogen and puts on an oxygen and the hydrogen there, making acetic acid. Which is now, this is much more water, acetic acid is much more water soluble than ethanol, and your kidney can get rid of it. And it's no longer ethanol, so it doesn't make you drunk. Now, has anyone ever heard of moonshine making you go blind? Okay, what's the deal with that? Well, moonshine is made distilled alcohol, usually from things that are fermented from fruits. A lot of fruits, when they're fermented, methanol, in addition to making ethanol, there's also some methanol made. Methanol is another alcohol. Looks very similar. You can see there's this COH. Instead of there being two carbons, there's just one carbon. Otherwise, it looks very similar. Now, when alcohol dehydrogenase acts on methanol, it turns it into formaldehyde. And formaldehyde is particularly toxic on the nerve and the eye, and that's why methanol poisoning, one of the effects of that is blindness. So if someone, so get your moonshine from a reputable distiller, otherwise this could be the problem. The other thing, everyone heard of the alcohol flush? Like redness that some people will get. It's very common in traditionally in East Asian populations. In those groups, the problem there is this enzyme aldehyde dehydrogenase doesn't work very well, or they don't have very much of it. So what happens is that ethanol gets converted to acetaldehyde, but then it stops there. And the acetaldehyde is what makes people really red, and it just makes them feel generally quite sick. It's not a very pleasant chemical to have. Usually it's quickly converted to acetic acid and gone, but in people who have a dysfunctional enzyme here, it doesn't happen. Side note, some of the treatments for addiction to alcohol, they'll give a drug that slows down the function of that enzyme so that this builds up and makes them feel sick when they drink the alcohol. So that's how that's working.
You can talk about how antibiotics affect all this.
I'm probably going to talk about antibiotics when I talk about grapefruit juice. Hopefully, and we can maybe have questions about antibiotics at the end if that works. Yes, sir.
Well, it seems that the liver comes in contact with virtually anything that's put into the body.
Yes.
So I'm wondering, when drugs are produced or manufactured, whatever, are experimented with, if the liver has also got to be a prime concern. I mean, it could be affected by any of these things, correct?
Yes, the question is, it seems like the liver affects anything that we take by a pill, because the blood's going to go through there. So is that something that researchers consider when they're designing drugs? And the answer is yes. They'll make like a type of the liver that works in a test tube. It's an extraction from the rat liver, generally, that has all these enzymes. So they can measure how much the liver is going to break down the drug. So we can know that. And really all that matters then is if you have to take more or less of the drug. If it's something the liver breaks down a lot of it, we just know we'll have to take more from the start to account for that. But I'll have an example that hopefully will explain that in a minute. Yes.
Josh, what that reaction you showed us with the liver.
Yep.
This is why alcoholism produces liver disease, and then liver has to work too hard.
So the question is this why alcoholism leads to cirrhosis of the liver? That's part of it. So the liver is actually quite an amazing, it's my favorite organ actually. It has a lot of interesting things. One of the things the liver does, it can dramatically change its size. And so if you use the liver a lot, it'll grow in size. So you can sort of overuse it, and that can contribute to the cirrhosis. There are other things too that damage the liver, but that's a major part of it. And then you're in trouble because if you damage your liver, everything you eat afterwards is not going to work right. The liver also controls glucose and lipid metabolism, does a lot of different things. So in addition to, another major class of enzymes that breaks down stuff are these enzymes called cytochrome P450, which we'll shorten to CYPs. There are 57 different types of CYPs in the human genome. Besides metabolizing drugs, they're also involved in synthesizing or modifying steroids, lipids, and vitamins. So it does a lot of other things in your body besides just breaking down drugs. These live mostly in the liver, but there are also some in the cells that line your intestine. That'll be relevant in a minute. There's also some in that blood-brain barrier I talked about. So it can break down the drugs as they try to get into your brain. Even though humans have 57 CYPs, just 5 of them metabolize 75% of the drugs that we take on the market. One of them, CYP3A4, metabolizes 60% of the drugs that are on the market. So a lot of drugs go through this same enzyme, which is going to create problems, as we'll see. So there's CYPs, these metabolizing enzymes are the major reason or underlying reason behind a lot of drug interactions. I'm going to skip over this. I was going to read a little case study that I use to teach my students about this concept. Basically, the gist of it is, this is a case study where a woman comes in, turns out she has AIDS, HIV, so they're going to give her a protease inhibitor that treats AIDS. And then, because AIDS targets your immune system, a lot of people will come down who have HIV or AIDS will have secondary infections, and in this case she gets tuberculosis. So they were going to give her a drug for tuberculosis, but it turns out that the drug for the tuberculosis, rifampin, increases the function of this enzyme here. So the drug that she was taking for her HIV gets broken down faster because there's more of this drug here, more of this enzyme there when she's also taking the tuberculosis drug. So it's just an example of how the drugs can interact because they work through these metabolizing enzymes. The other question is, what dietary interactions should be considered? And this is where the grapefruit juice comes in. So grapefruit juice, has anyone heard not to take grapefruit juice with their medicines? Okay, so we'll go through that. Here's an example. I didn't have it with the HIV thing, but there's a drug called felodipine. It's a calcium channel blocker, so it's used to manage hypertension, manages constriction of blood vessels and heart rate. That doesn't really matter for this. We have a graph here showing on the y-axis, this is the amount of the felodipine in your blood. And then the x-axis is the hours after you take the pill. So these open squares here show you the amount over time if you took your felodipine with water. So the concentration goes up a little over 10, maybe 12, and then it starts to go down. If you took your pill with grapefruit juice instead, the concentration goes up to 25, much higher. Does this have an effect? Yeah, because if you then measure the blood pressure, in water, when you take the blood pressure goes down here, whatever these open dots are, around 115-ish. But if you took the felodipine with grapefruit juice, the blood pressure drops lower. And so that could, because there's more felodipine, it's more functional, so your blood pressure could get dangerously low. So why is grapefruit juice causing this problem? So let's chart how felodipine gets in your body and moves around the body. So let's say here's your felodipine dose, your pill you took. There's 100% of the drug. It's coming down your esophagus, enters into your stomach. The pill just gets broken down here, so there's still 100% of the drug there. It leaves your stomach and goes into the small intestine. Okay, this is where it's going to be absorbed, right? So if we zoom in on the small intestine, we have here, this is the lumen. 100% of the drug is in the lumen. Remember, it has to pass through these cells to get into the bloodstream. Well, this enzyme, the blue dot represents that CYP3A4 enzyme. Some of that lives in these cells. So just by, in the process of passing through these cells to get into the blood, 70% of the drug is metabolized right there. It doesn't even have to go to the liver. Okay. Now it goes to the blood, gets into this vein. Remember we said the next stop for things absorbed from the intestine is actually the liver. So now it goes to the liver. If we zoom in there, so this would be the 30% case here. So that's the 30% that made it through the intestine. Now there's more of that CYP3A4 in the liver, and so that gets taken down another 50% to a final of 15% of what we actually started with. So then what leaves the liver going to the heart is 15% of the initial dose. And we know this, and we know that 85% of the drug that we take is going to be broken down. So we know what we want in the blood, so we give the right dose so that when 85% is taken away, we have the right amount left. So that sort of answers your question about how we account for the liver in drug design. Now, grapefruit juice contains a flavonoid which turns that CYP3A4 enzyme off, almost blocks its activity. So if you have this 100% here, in the normal case, it goes through this intestine, 70% is metabolized. But if you took that with grapefruit juice, this enzyme's turned off, now 90% of the drug gets through it. And then when it goes to the liver, the grapefruit juice didn't make it that far, create a problem. So it's still halfway cut down, but half of 90 is 45%. So now, leaving the liver, you have 45% of whatever you took instead of 15%. So when you take grapefruit, your pill with grapefruit juice, it ends up, the result is there's much more getting into your bloodstream than you would have had without the grapefruit juice. And since CYP3A4 metabolizes 60% of the drugs on the market, that's why it's a real problem to take grapefruit juice when you're taking almost any medication. It's the devil's elixir as far as pharmacology is concerned. Yes, sir.
Normally we wouldn't take the drug with grapefruit juice, but I've been told you shouldn't drink grapefruit juice within an hour or within 2 hours or something that is.
Yeah. It doesn't even have, I think in terms of the design of this experiment, the grapefruit juice was actually taken 30 minutes before the pill. But yeah, those flavonoids in there are sticking around. They can have an impact for a range of time. Yes?
This is grapefruit juice irrelevant.
Okay.
I want to know what the effect is of coating a pill and where it might be absorbed in the body instead. Does it only protect the esophagus, or does it do something further down?
So the question is, if you coat a pill, what's the effect of that? And it can be many things. So there's, you've heard of like long-acting versus quick-acting drugs. Sometimes you'll coat pills in ways to make them break down more slowly so they get absorbed more slowly over time. So it has sort of a longer effect. Other things are coated in ways that make them break down really fast, so they're really fast-acting. There's a lot of things that are mixed in with pills that don't produce the pharmacological effect, but they help either with absorption or something. So those are all the things that are in the pill. But yeah, it can have an effect depending on what the designer wanted. There's a whole branch of researchers who study those formulations. How do you formulate the pill to make it do what you want it to do? Now I was going to go through, I don't have time for this, so I'll see if I can bring it into our discussion, beginning of our discussion next week. In addition to drug-drug interactions, metabolism is where genetics can affect us a lot. So if I mentioned that there are these 57 CYP enzymes, there are variations where some people may have a CYP that's really fast-acting or slow-acting compared to sort of like the average CYP. So these things are heritable because they're genetic. And so there can be a genetic basis. If you have this mutation, you'll have more or less of the drug, depending on how the mutation works. So the metabolism is a place where genetics comes into play. I don't really have time. I don't want to speed through that because I don't think it'll make sense, and I want at least an interesting discussion. So we will move that to next week. And then just sort of the quick review for today. We remember we talked about drugs, how they have to bind a target in the body, but they have to get there to do that, so they have to be absorbed and distributed. Poor absorption is a major killer there's a lot of drugs that pharmaceutical companies have discovered. They say, oh, if we could only get it into the body, it'd be great. But we can't have it both ways. We can't design it so that it works and gets into the body. So a lot of things are killed, and because you don't want to take an injection for a headache, no one's going to do that. Because pills are the main way to go. A lot of drugs just die there. Once in the body, they're spread all about. Drugs are metabolized from the moment they enter the body. And then a lot of drug interactions and genetic differences, which we'll come back to next week, center on these metabolizing enzymes. So we'll end there. Looks like we have a few minutes for more questions if you guys have any.
Years ago, I was diagnosed with an onset of arthritis in my right elbow, and a pill was prescribed, and I asked for it to be given to me as a salve, and I used it, and the arthritis went away. And months later, I was diagnosed with arthritis in my left shoulder, and again, I asked for the salve, and within a couple months, it went away. And now I'm taking it for my right knee. This is metabolism. And if it had been a pill, it would have been a lot less potent when it got to where it was meant for.
So the story here was he was taking something for arthritis in his joints, just in one elbow or something. So rather than taking a pill, he had a salve that would rub right on that joint. And the idea was, so the thing is, you could take enough of a pill to get the concentration high enough in your elbow to be functional, but because once it enters the blood, it goes everywhere, that means you'd also have a lot in other places where you didn't want it, right? So this allows you to target that. It doesn't mean some won't get into your bloodstream and spread around, but it'll be much less. So that's an example of how people would try to limit side effects. You could take it via a pill, maybe that's easier than messing with the salve. But then it goes everywhere. We can keep the drug more localized that way. That's one way we try to limit side effects of things.
But why would a doctor prescribe it as a pill?
Mostly, so why would you describe it as a pill knowing that the ointment or the salve does that? And mostly, again, it comes back to the convenience thing. If you just tell a patient, here, take this pill once a day with breakfast, that seems so easy that that's what most people gravitate to, where, oh, you have to rub this on 3 times a day. It just seems more work, and some people might not want to hassle with that. Which I think is unfortunate, that a lot of prescriptions sort of come down to that, what's easier for the patient when maybe it's not better, but if they won't do it, if they won't rub it on, or they keep forgetting or something, then it's not having an effect either. So those are the trade-offs. Yes?
What causes drugs to become less effective over time?
Oh, you mean sitting in the pill, like in the box or the bottle?
I mean some that you've been taking
for
months and years, and they don't seem to be as effective after a period of time.
Yeah, so the question is why would drugs that you've maybe taken over a period of years lose effectiveness? There's a variety of answers to that. It depends on the specific case. But one example is, so let's say you take a drug that's metabolized by a certain enzyme, and we know that enzyme is going to knock out 50% of it. Well, our body is quite adaptable, so if it notices, hey, this enzyme is getting used a lot, maybe I should increase the amount of the enzyme that's in my body. So your body will up that, which means more of the drug gets broken down, and then it may get to a point where it just gets broken down so fast, it can never be effective in the body. So that's a specific example of the general phenomenon of your body's adaptable and it might make some change that then makes the drug no longer work.
Josh, thank you so much for Don't forget to come back next week for part 3. Before you leave, please remember to pick up your coffee cup from under your chair. And if you feel that you are able-bodied to help us put away chairs, do that as well. Thank you all. We'll see you next week.